So primary visual cortex is the earliest cortical area devoted to vision. It's at the back of your head, an occipital cortex. And here's a diagram showing the visual pathways before primary visual cortex, or V1 as it's often known for short. Here's the visual field, and they've divided it into left and right. Notice that the right eye can see part of the left visual field as well as the right visual field, and similarly for the left eye. And input from the two eyes are segregated so that information from the left visual field ends up in primary visual cortex in the right hemisphere of the brain. So you'll see V1 on each side is receiving input from both eyes, and the same is true of lateral geniculate nucleus, the lateral geniculate nucleus of the thalamus, which is a relay station, if you like, on the way to V1. But in the thalamus, information from the two eyes is kept separate. It isn't really combined at the level of cells. Information from each eye is segregated into different layers of the thalamus. And it's really only in V1 that we start to find neurons that are clearly receiving input from both eyes. So there's a first area where you find neurons with binocular receptive fields, meaning a receptive field in each eye. And many of these neurons are tuned to disparity. So what I mean by tuned is that their firing rate modulates as a function of the disparity between the images. Now these curves are obtained with what are called random dot patterns. So it's a little bit like the disk image that I showed you earlier. So it's a cyclopean stimulus where the left eye is just seeing a pattern of black and white dots on a grey background, and the right eye is just seeing a pattern of black and white dots on a grey background. But within the receptive field of the neuron, the dots in one eye are shifted with regard to the dots in the other. So that's the disparity that's plotted here on the x-axis. And the dots and the error bars show the mean firing rate of the cell as a function of the disparity of the random dot pattern. And these plots are typically averaging over many different random arrangements of dots, but all of them have the same disparity. And you can see that the cell is very sensitive to disparity. So this cell, for example, is firing at 25 spikes per second for this disparity of sort of minus 0.6 degrees, whereas when the disparity is, minus 0.1 degree, the firing rate drops right down to only about 5 spikes every second. The horizontal lines for comparison are showing the response to a dot pattern presented in the left eye only, or in the right eye only. And you can see that if you consider monocular stimulation, this cell looks as if it's responding only to input in the right eye. So you're getting about 27 spikes per second when a random dot pattern is displayed in the right eye, virtually nothing when it's displayed in the left eye. But when it's displayed in both eyes, you're getting this complex firing rate that varies depending on the disparity. And here's another example neuron showing the same thing. Now, many of the properties of the camouflage breaking cyclopean stereopsis seem to be set by the property of these neurons that we find in V1. And that's led to the idea of V1 as being the cyclopean retina. It's a term that was first introduced by the Hungarian psychologist Bela Ules, who I mentioned earlier. So his idea is that just as your physical retina is where your body first receives the light signals that ultimately result in vision, he's suggesting that V1 is maybe where you first begin to extract the cyclopean information that can only be obtained by comparing the two eyes' images. So if you have a neuron that's binocular, so by definition it has a receptive field in both eyes, that's why it's called binocular, the receptive fields of V1 neurons are pretty small, so it's only seeing a little patch of each eye's image. And you can only perceive a disparity if you have a neuron with the appropriate receptive field with that disparity. So what I've tried to sketch here is on the left we have the left eye's image, on the right we have the right eye's image, and you have all these different V1 neurons, these little pink things here, with receptive fields at different locations in the two eyes. And you can see all these neurons, they're differing both in the location of the receptive field, but also in the disparity. So this neuron here has got quite a large disparity. So if you imagine that these are the entire retina, then the left eye's image is quite close to the left-hand side, whereas the right eye's receptive field is shifted quite a bit. Whereas this neuron has got much smaller disparity. The receptive field's at roughly the same location in the two retinas. And you need a receptive field, you need a neuron whose receptive field is tuned to a particular disparity in order for your fine cyclopean stereopsis to be able to perceive that disparity. At least that's our current belief of how, the neural basis for stereoscopic vision. Now actually most V1 receptive fields, when you measure them, they're actually at rather similar positions on the two retinas. So they're typically within around plus or minus 0.5 degrees of one another. So if you have one neuron where the left eye's receptive field is, say, 10 degrees from the fovea, the right eye's receptive field, it might be 9.5 degrees from the fovea or 10.5 degrees from the fovea, but it won't be 5 or 20 degrees from the fovea. So most V1 receptive fields are tuned to rather small disparities, and that seems to be why we can only see or fuse disparities which are near zero. And as we saw before, outside this range, we see double. You can't fuse the images anymore. And we think that's probably because you don't have the V1 neurons that are tuned to such large disparities as you get when you separate the objects in distance. I now want to introduce something called the correspondence problem, or more precisely the stereo correspondence problem. So I defined disparity using this diagram, and I talked about the difference between the image of the apple and the difference in position between the image of the apple and the image of the orange. And that worked because we had very different objects here. You can clearly distinguish an apple and an orange. But we also saw that dot pattern, do you remember, with the white squares on the black background? And that seems much more challenging because now all the little features in the image, all the little squares, are indistinguishable. So if you have a retinal image with a bunch of little squares, how do you know what visual scene has caused that to fall on your retina? It might be this, but it might be this, it might be this. Basically, you could have a square at any of these points where the lines intersect. So how do you know what's really present out there in the world? Perhaps surprisingly, we don't struggle with this at all. In this situation, pretty much everyone is always going to see this solution, which indicates the smoothest surface that's sufficient to explain the retinal images. So your brain's very good at solving this correspondence problem. And that's a bit of a problem because individual V1 neurons, as we've seen, are just seeing different, very small patches of the left and right images. So one neuron might be seeing this pair of patches, and the next neuron along might be seeing this pair of patches, this one. And of course, it's this V1 neuron that's seeing the correct match in the sense that it's seeing the same thing in both its receptive fields. So this neuron is seeing the disparity of the scene here. And I'm going to refer to that as a match. All the other neurons, they're not seeing matching parts of the images. So they're tuned to disparities, but those disparities are not present in the scene. Only this disparity is present in the scene. So how do you know, if you're the brain, which neuron is seeing the true match? Well, a very simple answer is simply to cross-correlate the image patch in the two windows. And what I mean by that is take all the pixel values in the left eye's patch and in the right eye's patch. So like at the centre of the image in the left eye, we'll plot that along the horizontal axis and then plot the value at the centre of the right eye's image and so on. And by value, I mean points where the image is dark are going to have low values, and points where the image is bright, like this white here, will have high values. So if you do that, you just, for each location in this little circular patch, you take a location in the right eye, plot that along one axis, same location in the left eye, plot that along the other axis, and you get this scatter plot. And you can see how well those pixel values agree. And you can see here the answer is not very well at all, because obviously the image patches are completely different. But when the image patches match, then the correlation is 1, because wherever you are in this little image patch, the left eye and the right eye's patch are always identical. So your brain's faced with a problem. You've got all these neurons with their little pairs of windows into the two retinae, and you need to work out which of these neurons is looking at corresponding image features. So let's say you choose the group of neurons which all happen to have the same receptive field location in the left eye, so I'll keep this window here constant, and we'll look at a bunch of different neurons whose receptive fields in the right eye are at slightly different locations, and we'll figure out which one is viewing the corresponding image feature. Or we can do that by plotting the correlation of the image patches observed by each neuron. So this particular patch has quite a low correlation, as we saw, and then we'll move through, and there'll be a very low correlation, say for this point here, but by the time we get over to this point, then the correlation will be 1. And so you, as your brain is looking at all these neurons, trying to figure out which one is looking at the correct match, you can deduce, this must be the stimulus disparity, because neurons with this preferred disparity are seeing the highest correlation. So that's a sort of overview of what we think is going on in VU1 to compute disparity. So interocular cross-correlation like this is referred to as a goodness of match metric, and the nice thing about it is it works on absolutely any image. So the example I just showed you with that statue had recognisable image features like someone's head, the eyes of the statue and so on, but it can also work just fine for random dot images, these cyclopean images like the disc example we saw earlier, because once again you can, as it were, slide a little piece of the left image over the right image until you find the point where it matches perfectly, and so the correlation is 1, and therefore you can deduce the disparity between the left image and the right image without needing to recognise any objects within either eye's image. Now it turns out that if you compute cross-correlation within these little local image patches like that, that also automatically gives you a preference for smooth surfaces. So it gives your visual system a kind of bias towards perceiving these smooth surfaces, and that's quite useful because the world tends to be made up of objects where a distance is generally changing rather slowly. It may be changing because the object's slanted with respect to your point of view, but in general you don't see clouds of random points like we when I showed you some of the other solutions to this matching problem, they were kind of clouds of random points, and that's really not what visual scenes tend to be made up of. They tend to be made up of discrete objects across which disparity changes relatively slowly, and then there are these sudden discontinuities at object boundaries. So a preference for smooth surfaces is quite useful, and that seems to be built into our visual system through these V1 neurons that look for disparity within these little patches.